Hey guys, we are on the third part of the TEAS science review now. We're going to talk a little bit about earth science, some physical science, and a little chemistry. So I'm going to jump right into this section and talk about, again, the concept of energy. And the energy molecule that our bodies use is going to be ATP. So when we break down food and we harvest energy from it, this is where the energy comes from, it is this molecule right here, okay? Adenosine triphosphate, ATP. So that's really important to know right off the bat for energy. Two main types of energy are going to be stored energy, which is basically, we'll put it here, potential energy. Okay, it's like water behind a dam, getting ready to be used but not used yet. And of course, the energy of motion is going to be kinetic energy. That again is the energy of motion. So when we think about, now those are the different types of energy. Now, when we talk about chemical reactions, it's a little bit of the chemistry part of it, but we want to understand this. An exergonic reaction is going to be a reaction that is basically, we have some other terms that we could use for, like catabolic or oxidation. It is simply a breakdown reaction. So this would be the example of eating food. What happens inside our bodies when we eat food is that we break it down by exergonic reactions, okay, to harvest ATP. So this is how we basically gain ATP, okay, is by these exergonic reactions. The exact opposite of this type of reaction would then be the endergonic reaction, okay, which would be the same thing as an anabolic, okay, or reduction type of reaction. Okay, there's different terms for these, but the endergonic reaction would basically be, you could throw in another term because this describes it well, a synthesis type of reaction, okay, and in this case, we're gonna build. This is a building type of reaction. This is what our bodies do, for instance, when we make proteins inside the body. We would be making proteins, okay, and in order to do that, we're going to basically need ATP. So put another way, right, these reactions basically use ATP. They burn up ATP. Our cells have little sodium potassium pumps. They would use ATP. So talking about these reactions and talking about energy again, let's talk about something called active and passive transport. Passive transport uses no ATP. So it uses no ATP. Okay? So I'm just going to throw this in. This is kind of biology, but it is important to understand. It kind of is still along these same lines. Passive transport would be transport through the cell wall, okay, and it would be no ATP necessary. And so I just want to throw this out there. It's going to be things like osmosis, okay, and it's going to be things like diffusion and things like filtration, okay. Diffusion, different things. We've used the term diffusion before. We're talking about oxygen. Oxygen diffuses into cartilage from the underlying tissue. Same thing with the epidermis. There's diffusion of oxygen from the underlying dermis. Okay, so diffusion is simply the movement of particles or substances from a high to a low concentration. Okay, osmosis is really important to understand. It is simply the movement. This is abbreviation here for movement of water. That's what's really important to know about osmosis. It's the movement of water, okay, through the cell membrane. Again, it's going to basically go, in this case, it's going to be attracted to the higher solute concentration. So for instance, if there's a lot of sodium in the cell, water is going to be attracted to that sodium in the cell. If there's a lot of sodium, to use an example again, sodium outside the cell, it's going to be attracted, or the water is going to move basically to the outside of the cell. Okay, so the key thing to remember though about osmosis is it's movement of water through a permeable membrane, through the cell membrane. Filtration is going to basically occur, for instance, in the kidney. It's going to be basically movement through the walls of basically the capillaries, which acts as a filter. Okay, so we just want to know that these are the three types of passive transport. Now the opposite of this, I'll put it over here, is active transport. And the best example that I will use is the sodium-potassium pump. And the sodium-potassium pump would need ATP. Okay, would need ATP. So that's an example of active transport. It's going to be any process of transport that requires ATP. Okay, so keep that in mind. We're just kind of trying to knock out some of the important parts, important info here. Now, moving along, I'm just going to mention really quickly what we call the tonicity of the cell. Tonicity, okay, let me erase some of this real quickly. We kind of talked about this. We talked about the cell shrinking and swelling. Excuse me, hypertonic solutions will typically cause shrinking of a cell, and it basically shrinking of a cell. And the reason for that is it goes back to the same principle we were referring to with osmosis. If I am to drink a bunch of salt water, that's going to create a hypertonic environment in my body. That's going to cause all the water, and that's because all the salt from the seawater is going to be basically outside. It's going to be in the extracellular environment. It's going to be outside my cells. So all of that water in my cell is going to go where? It's going to go toward the sodium. So that means it's going to leave my cell and shrink it. Another term for this is crenate. Okay? Therefore, the exact opposite will occur with a hypotonic. Make sure I spell that right for you. A hypotonic solution. The hypotonic solution will cause the cell to swell, and a perfectly balanced is referred to as isotonic. So, no wonder, here we go. Isotonic solution will cause basically no change. This is a perfectly balanced environment inside and outside the cell. Okay, so that's what we refer to as the tonicity of the cell. Now, moving on, talk a little bit more about chemistry. We're going to get into what we call inorganic chemistry for just a minute. Inorganic chemistry refers to, the term inorganic refers to without carbon. Without carbon. So if something's inorganic, it contains no carbon. Inorganic chemistry involves our periodic table. So we look at the table, we see all these boxes with abbreviations inside that represent elements. So again, the periodic table is basically a table of all of the elements, and every element is going to be inorganic because they're all without carbon. They're actually in their simplest forms, okay? Which means they can't be broken down any further. They're as simple as they get. So they only basically contain an atom of whatever, hydrogen, or they're dealing with this one here, He is helium. We're going to use that as an example, but none of them contain carbon, so everything on the table is an element in their simplest form, referring to it as, we can refer to each of them as inorganic, okay? Another thing we can refer to is minerals. Elements are minerals, and we know that many minerals our body needs. Some we don't need. In fact, some are toxic on this periodic table, but we get it from the earth. So this first, I'm going to give a quick example of its helium. And helium has, on the periodic table, everything has a number here. This number, and I'll use a different color here. This number is the atomic number, which is the number of protons, okay? If we go down to the bottom, We also see a different number. This is going to be referred to as the atomic mass, which is the weight of the atom. How do you calculate weight, or how do they calculate it? It's easy, actually. It's simply our protons plus our neutrons. Okay, so protons plus neutrons, we add them up. That gives us the atomic weight, or mass. Here's our atomic number, again, which is simply the number of protons. Okay, so what does that look like? If we look at an atom of helium, this is going to be our atom of helium. Atoms have subatomic particles, okay? This is going to be the nucleus of the atom. Now, inside the atom, we would have subatomic particles. We have the proton, that's a subatomic particle, which has a positive charge. We have the neutron, which has a neutral charge, not negative, neutral charge, okay? And then we would have circling in what they call concentric circles. It's more like clouds around the nucleus, is the electrons. Notice the electrons have a negative charge. So if I want atomic mass, I'm going to add up protons and neutrons. If I want atomic number, of course the table tells me, but I would add up the number of protons. We know helium has 2. Now, electrons are really important because they're going to deal with, especially the outer row of electrons called valence electrons, they have a lot to do with the bonding properties of one atom to another. Now, the other thing that's really important about electron is it has a negative charge. So if I'm looking at an atom, let's say, of sodium, sodium contains a positive charge inside our bodies. So we're looking at sodium. What do I want to look at to know if it's going to have a positive charge or not? Well, I would look at protons because they have obviously a positive charge. I'd also be interested in knowing the number of electrons because they have a negative charge. And in this case, because sodium always has a positive charge inside our bodies, I would know that they would have to have more protons than electrons. Okay, why? Because the overall charge is positive. So I don't know the exact number. I think it's around 11 or 12. So for instance, there's probably, let's just say hypothetically, 12 protons and, for instance, something like 11 electrons. There's got to be more because it has a positive charge. If I looked at something like chloride, it would have to be the exact opposite. It would have to have more electrons than protons. Why? Because chloride always has a net negative charge in the body. So keep that in mind when we're looking at charge. We're only concerned with protons and electrons. We don't care about neutrons because they're neutral. They make no difference. Okay? Why do we not care about electrons for weight? Because they have very, very little weight. So we don't count electrons. We only count protons and neutrons when it comes to weight. So that's a little discussion on the periodic table. Now, if we were getting into bigger molecules, okay, a lot of which we call polymers, we would be discussing something called organic chemistry. So the term organic would simply mean with carbon. Organic chemistry is always going to be larger molecules, okay? In fact, when we talk about macromolecules, which again are large molecules, they're also referred to as polymers because polymers have repeating subunits. We're generally dealing with 3 big, really we could say 4 big categories, really, but 3 main ones, and that would be our carbohydrates, our fats, and our proteins. So typically, we're dealing with organic chemistry. We're dealing with these larger molecules. We're discussing carbohydrates, which have repeating subunits, particularly when we get into the polysaccharides. They would have repeating monosaccharide subunits. Fats, right, have fatty acids. Proteins, of course, have amino acids. So keep that in mind when we're dealing with organic chemistry. We're dealing with these larger molecules. Okay. Another thing I'll mention when we're talking about this is enzymes. Now, anytime we're dealing with an enzyme, we typically see that it ends in ASE. That's a little trick. So it could be lactase. That tells us that it ends in ASE, so we know it's an enzyme. Not necessarily we don't know what it does, but what do enzymes do? They're going to help allow reactions to occur, okay? So keep that in mind when we're talking about this organic chemistry. Enzymes allow reactions to occur, okay? Okay, so moving on, that's all I'm really going to say about enzymes. I want to talk about acids and bases real quick in our pH scale. We're dealing with a pH scale, really dealing with a scale based on 0 to 14, 7 being right smack dab in the middle, so that is neutral. The only thing naturally occurring that's neutral is water, okay? Therefore, anything below 7, not 7, but below, right? So literally 6.99 down is an acid, and above 7 would be bases, okay? So for instance, a 6 is an acid, but the further I go down in number, the stronger the acid. So an acid of pH 1 is much stronger than an acid with a pH of 6, okay? Same thing with the bases. If I have a pH of 8 versus a pH of, let's say, 13, the 13 is going to be a much stronger base. Acids release hydrogen, okay? They have a lot of hydrogen and they release it when they react. Whereas the base, so we could say over here that it releases the hydrogen. The base accepts it, okay? So they're going to be lower in hydrogen. This pH scale is really based, therefore, on hydrogen concentration. The lower the number we go, the more hydrogen we will see, okay? The higher the number, the less hydrogen, okay? Another thing that we'll mention is every time we change a number, whether we go up or down, in this case we're going to go down in numbers, we're going to go from 7 to 6, that increases the strength, in this case of acids, 10 times. So we're going to go 10, so meaning a 6 would be 10 times as strong an acid as a 7, which is, of course, we said is neutral. If we went down, let's say to like a 4, that would be 10 times 10 times 10. So 4 is 1,000 times stronger an acid than a 7. Okay, if we went to 3, that would be 10,000. A 2, 100,000. So you get the point. And the same concept with the base. Okay, so that's the basics of the pH scale that you will more than likely see on the TEAS test. You know, I can throw in one more thing that is beneficial, dealing with the term saturated and unsaturated. You hear this a lot with fats, saturated and unsaturated. And the term is very simple. Let's say it's again fat. Saturated would simply mean, and again, when we're dealing with fats, we're dealing with carbon, right? Because we're dealing with organic chemistry. So to use the example of a saturated fat, we're dealing with, didn't give myself a lot of room here, but we're dealing with basically carbons. Let me, this is the saturated still, give myself some more room. We're dealing with carbons that are literally surrounded by hydrogens. What we call it? We call that saturated. They're saturated. Versus if we were dealing with, let's say, carbons that had some missing spots. Maybe it's something like this. I'm just kind of making something up here. This would be unsaturated because it has open spots. The hydrogens do not completely surround the carbons. That's really where the term comes from. Okay. We know there's many different types of fats. Keeping in mind this too, we've got saturated and unsaturated, right? So we showed you the saturated versus the unsaturated. Saturated fats typically are found in animal products. So you can kind of remember that versus unsaturated are again typically, say 99%, are found in plant-based products. So keep that in mind. So something like vegetable oils. A lot of people say, well, vegetable oil is not real good for you. So be it, but it is an unsaturated fat, okay? versus saturated fats are going to be things like animal-based oils, fats, okay? So that's just keeping in mind there. Another thing that we could throw out too would be the thing called trans fat. If we drew a trans fat, we're basically drawing fats with hydrogen and oxygen that are basically opposite each other, right? Trans. You can read my writing here. Trans, right? Trans meaning across. Versus what we call a cis fat. What I just mentioned this because there's a lot of discussion on it. Trans fats are basically man-made processed fats that are basically considered not good for you. Cis, meaning same side, okay, naturally occurring fats. So I mentioned that is in the anatomy textbooks now. So I just mentioned that. The other thing that we can kind of wrap up with here real quickly would be bonds, chemical bonds. We have the ionic, we have the covalent, and we have the hydrogen bonds. Okay, these are all bonds that we see in the body. The ionic bonds is always going to be between ions. Good example is going to be it's going to be a bond between a positive and a negatively charged ion. We call the positively charged, these are ions because they're charged. We can go a step further, we can call the positively charged a cation. We can call the negatively charged an anion, okay? And again, naturally occurring in the body, these are going to want to bond. So when they bond, of course, in this case, we get NaCl, sodium chloride, which contains an ionic bond. It's a weak bond. It breaks up very easily, okay, but it is nonetheless a bond, okay? Now, a different type of bond, which is actually a stronger bond, is going to be the covalent, okay? The key thing to remember here is this is a shared bond. Shared between what? Electrons, okay? It's the sharing of electrons. So if you see the word covalent, you remember shared. Come down to hydrogen. Hydrogen bond is going to typically be between a positive hydrogen and a slightly negative oxygen. Also a weak bond, okay, but very important bond in the body. We see it with proteins and we see it with DNA. And we also see these bonds with water molecules. So this is a very important bond as well. Again, these are going to be the three main type of bonds that we're going to see in the body. So just a quick rundown on chemistry. There is more to come. We'd like to do some more microbiology type topics. But stay tuned and good luck, good studying.